Hi. Today's topic, I didn't want to talk about it because it's already everywhere. Everybody talks about it, and also it's a very actual topic. So actual that a lot of things that are not completely clear yet. But I realized that a computation about evolution in natural cell action without a discussion about molecular clock is lacking something. So let's go with the molecular clock. We say that DNA mutate in time and we just ignore the reason it happens. We can imagine for now that it's due to some error during replication or some UV rays. It's not that important, the reason. Thing is that it mutates in the way that if you start in a moment of time with a sequence, after some delta time, that is just a fancy way to say that some time is passed, you will have a mutation. A letter of the DNA sequence is changed. If you wait another delta t, two letters will be changed, and so on and so forth. The rate of mutation is, I'm lying to you, but let's imagine that is nearly linear. It's a lie, okay? But we will talk about it later. So we can imagine that DNA mutate at a constant rate. We will dig into this concept, but just for a minute, bear with me. If we know that a mutation occurs, every 10 years, I know that if I take two sequences that differ for two letters, they are evolutionarily separated from 20 years, because 20 years of separate evolution are needed in order to give rise to two mutations. So now let's be realistic for a moment. DNA don't always mutate linearly over time. Let's imagine the situation. Let's imagine a chunk of DNA that codes for a pivotal protein, a protein that carries an important role. A mutation in this protein will be really dangerous, maybe making the protein useless, and the organism with the mutation will die without passing its mutated DNA to the progeny. So a mutation in this kind of DNA pieces will occur very slowly. They need a secure environment in order to don't be lethal. Now let's imagine the opposite situation, a mutation in a piece of the DNA that is useless. The organism with the mutation probably will not even notice the change in his DNA letters. So a mutation in this kind of DNA sequence will propagate in the population, and mutation in useless DNA will very rapidly accumulate. So if I take the 3 billion letters of the human genome, not all these 3 billion letters will have had the same mutation rate over time, from 3.5 billion years ago and now. 3.5 billion years ago was the moment the life arose in Earth. Because every chunk of DNA serves different role. The same reasoning could be done over DNA chunks of different species. They also mutate at different rate. A chunk of DNA that in human mutate at a slow rate, maybe in a fish mutate very rapidly, because they serve different role in humans and fishes. The first example that came to my mind is prolactin, this protein here. Prolactin is a proteic hormone that regulates the milk production in humans, in female, but that regulates skin change in tritons. So there is this other component that add some complexity, some imprecision in our calculation with the molecular clock. In the first slide, we have given for sure that our delta t is always the same, the time that a mutation occurs, but this is not always the case. Now, let's look at how scientists have thought about resolving this problem. They said, we need to find a chunk of DNA that serves the same role in all the critters in order to make the mutation rate similar between different species, and we need a chunk of DNA that mutates so slowly that it could give us information about million or billion years ago, because this is the time scale we look at in evolution. So it wasn't an easy task, but finally they find a DNA chunk that fit the requisite. they find this chunk of DNA. This chunk of DNA that code for a specific product, an RNA called rRNA16S, that stands for 16 Svedberg filament of ribosomal RNA. It's mouthful and useless to repeat it, but the important thing is that this chunk of DNA is conserved. It's conserved in all the organisms because this rRNA16S is really important. Okay? It's a part of the ribosome nanomachine. We've talked about it when we talked about central dogma. It's necessary to translate RNA into protein. So it serves the same role in all the organisms, and it mutates very slow because a mutation in the ribosome is almost always lethal, because an organism with the wrong mutation in this machinery will not produce protein, so it will die. And it mutates very slow. So they started to look at the DNA sequence of this rRNA16S, and they found that every organism has a mutated form. Every organism has a mutated form. It means that all organisms have different rRNA16S sequences. That is wonderful because it is what was expected. The interesting thing that was also noticed was that the difference between rRNA16S sequences were proportional to the years of separation between species. In other words, a separation between species a long time ago allowed this chunk of DNA, the rRNA16S sequence, to differ a lot. And instead, a relatively recent separation between species will result in a very similar rRNA16S sequence. So I can sequence now a lot of these rRNA16S from a lot of species and measure the difference in letters between each other. So I will know, in function of the difference in sequence, which species are near in the evolutionary tree and which species are very far. That means they separated a long time ago. So I can build an evolutionary tree, but it's not finished here, the potential of this concept, because okay, now we know in what order the species separate one from the other in a relativistic way, but we know that bacteria separate from eukaryotic before bonobos and chimpanzee, separate one from the other, but for now we haven't talked about timing. We have a relative idea of the order, but not a very clear idea of how much real time is passed between the separation of bonobos and chimpanzees, or every species on the earth. And here we utilized the concept of constant rate of change of the DNA. If we read two sequences that differ for 4 letters in the DNA chunk of rRNA16S, and we know that in this chunk every letter changed like one time in 1 million years, we know that these two species separates like 4 million years ago, because they differ for 4 letters. The formula is not that simple because it takes account of the fact that when the species separate, all the two species start to mutate, and not only one, so they will divide the number of mutation by 2 and other stuff, like the probability that a mutation is repaired from another mutation that make the original letter to return, this kind of stuff. But the idea is this: you sequence two species, you read the letters of the rRNA16S chunk in the DNA, and now you know more or less when they started to separate their evolution. So you know when they became different species. This is the way we imagine that humans and chimpanzees are the closest relatives in the primate world, and they separate about 6.5 million years ago. All this theory of molecular clock is of course confuted with the fossil records and precedent knowledge. And I wanted to repeat it, it's a young theory, it's not perfect yet, and sometimes the results are just strange when you confront them with the previous knowledge and fossil records. But it's a promising resource of information in order to investigate our past. and how humans became humans, chimps became chimps, and how I don't know how beautiful and tangled is our life with the life of all the other creatures on Earth. So this is it for today, and as always, let me know what do you think about the topic we've talked about. And I always put emphasis on that when I talk about topics like this, because they are, in my opinion, particularly interesting and offers a lot of point of discussion. So thanks for watching, and see you later.